Hello, good afternoon. Um, I want to welcome you to another edition of our educational series on Omega Bioedutainment. Today, again, we are focusing on general microbiology, and that is your journey as a beginner in microbiology. Today, uh, today's lecture is divided into two sections, just like I said. The first section is a pre-recorded message, pre-recorded of what are the basic materials that you can use whenever you want to cultivate or you want to culture uh, microorganisms. So that one is already a pre-recorded message. Immediately after this session, this theory session, I am going to upload that so that you'll be able to view that as well in line with this. So in that video, I was able to list out I was able to list out these materials, uh, laboratory materials for cultivating microorganisms in the lab. So, what are those essential materials that you actually need? So, I listed about 10 of them which are available. So, I want you to check out for that later on. So again, this afternoon we are going to look at types of culture media, the type of culture media and their uses. That is, what are the basic types of culture media if you want to classify culture media. Yesterday in the yesterday lecture, I told you what a culture media is. It's just like you are culture media is a kind of a food that you preserve. I mean, that you prepare for microorganisms to be eaten while you keep on uh, studying them in the laboratory. Don't forget that I told you that microorganisms is they are just too small to be seen with our naked eye. And if you want to see them alive, if you still want to see them alive, uh, you only see them if you are able to culture them. So don't forget what we said yesterday. I made mention that you should not forget your journey as well. Aseptic techniques is number one basic the number one basic prerequisite or basic characteristics or basic requirements if you want to be a good microbiologist. You need to maintain all your workings, all your samples. You need to make sure that you maintain them in aseptic condition. And I define aseptic condition for you as a procedure or a state whereby your work is not being contaminated by external factor. Okay, so then the next thing is your your power in microscopy and staining are very, very important. So that is microscopy and staining. The third one is culture method. So you should be able to know different culture method. That is the summary or review of what we did the last time. So yesterday we are able to touch on this. So on this culture method, I was only able to explain one or two things. And in all these conditions, we went as far as considering the inoculation or the incubation condition. So that we don't waste our time today, today is going to be for types of culture or classification of culture and their uses. Now let me try and list out the different categories that we have. The first one is general. It's called general purpose agar or general purpose media. Okay, that's the first one. The second one is will come under enriched media. That is very, very important. Then the third one is selective. I think we have like 8 selective media. After that, we have a differential media. So after that differential, we also have 5. We have transport media. Okay. Number 6, we have synthetic. We have minimal synthetic defined media. Okay, so the 7th one is semi-solid media. Then the 8th one, I said we have 9. The 8th one is what is called specialized medium or media. So we are going to see example of this and what are the factors that determines which one we are going to use for. Don't forget that each organism require different types of media to be able to cultivate them. Different types of media in order to cultivate them. So that is what we will be doing. We just start picking them one after the other. And what are those things that really determines how they can be used and the purpose of which they can be used or they are being used for. Don't forget we have 8. So if you want to remember that, just write DES, okay, DTM, and SS. So D is standing for, sorry, G, not G. G is standing for general purpose. E is standing for enriched. S is standing for selective. D is standing for differential. T is standing for transport. M is standing for minimal synthetic defined. I will tell you the reason why the name of this is very long. Minimal synthetic and defined. Okay, so the second to the last is semi-solid media, and the last one is called the specialized media. So we are going to see all this as we go on. We are going to start picking them one after the other. We are going to give description. We will describe them. We will describe them, and we are going to uh, see the reasons why we actually need them, the reasons why we actually need them, and the reason that determines. For transport, M is standing for minimal synthetic defined. I will tell you the reason why the name of this is very long. Minimal synthetic and defined. Okay, so the second to the last is semi-solid media, and the last one is called the specialized media. So we are going to see all this as we go on. We are going to start picking them one after the other. We are going to give description. We will describe them. We will describe them, and we are going to uh, see the reasons why we actually need them, the reasons why we actually need them, and the reason that determines. So I want to welcome you back to uh, picking types of media. We will describe them, we'll give examples, and what are the major factors that determines which one we are going to use. The first one we are going to look at is the purpose general purpose media. And just like the name suggests, general is okay, it is called general. The description is that number one, M is standing for minimal synthetic defined. This media contain the name of this is very long. Minimal synthetic and defined. And what are the major factors that determines which one we are going to use. The first one we are going to look at. So this is a media. This media consists of or contain. This media contain a balanced mix, a balanced mix of nutrients, mix of balanced mix of nutrients, balanced mix of nutrients. Okay, Nutrients like that contains peptones. After that, beef extract, and agar. Agar is the solidifying agent, okay, which are designed to support the growth of general microorganisms, to the growth of very wide microorganisms, including bacteria and fungi, okay. So typical examples we have in each case is a nutrient agar. This one is in solid. So because of the presence of agar, wherever you come across agar is the solidifying agent. So, or nutrient broths. Anytime you also come across nutrient broths, this one is telling you that is a liquid. So almost all the components, they have the same component. The only thing is that this one has a solidifying agent, which is called the agar, to make them to exist in solid, in powder. But this one is going to be broths, which make them to be in liquid form. So example is a nutrient agar. Let me try and bring example of a nutrient agar. Okay, and that is exactly what I did in the first section. That's what I did in the first section, where I basically gave you what is so this is an example. This is a nutrient agar. It's a general purpose agar. That is used to grow fastidious and non-fastidious microorganisms, including but not limited to fungi and bacteria. I've given you examples of this. So, and if you look at the components, you will see that this one contains peptone. It contains peptone. Peptone is going to be the source of protein. Beef extracts, and agar, which is the solidifying agent. So if you read here, you see that the ingredients, agar is 15 grams per liter. Peptone is 5 grams per liter, and sodium chloride. This sodium chloride is you see, salts will definitely there are many microorganisms that they cannot grow if they don't have salt. So microorganism so if you don't have anything in mind, you just want to culture, you just want to grow microorganism, so you are not so bothered about whether you want to use it for identification. If you want to use for identification, that is when you use a different agar entirely. So, but for general purpose, you just want to grow microorganism, so you make use of a nutrient agar, and the liquid form of nutrient agar is called the nutrient broth. So now, why would the purpose? Why do people prefer nutrient agar? I've given you that one of the first reason why we prefer using nutrient agar is just because we don't want to be specific. We are not so interested in identification. We don't want anything that is going to be complex. So what we need is we need it for simplicity. You just want to grow microorganism. What you use is a nutrient agar. That is why it is that. So number one, it is broad spectrum. It supports the growth of a wide microorganism. Okay, it is a broad spectrum. And number two, because of its simplicity. Please take notes. The two reasons why people will prefer the use of a nutrient agar, it is the one that is readily available. You don't need special skill. Once you know how to prepare it and you make it to solidify, what you need is you just need a nutrient agar. You just need to study microorganisms in their still existing form, that is when they are still alive. So what you will do is you make use of a nutrient agar. So that one will now lead us to the second types of agar, and that is enriched media. So from the word enriched, that is fortified. For example, now, if you know there are some gari, everybody knows that gari is a source of carbohydrates. Okay, but we now have fortified gari now that are already fortified with vitamins. So, for example, now whenever you are not taking your gari and you don't want to take it just raw like that, so what you do, you add granite, you add milk, you add other things that will make it that you are eating something that is balanced. So the same thing with enriched media. So enriched media is that instead of you to have a general purpose agar, you now want to enrich the nutrient agar with a source of other things, source of other nutrients. So that is enriched, and that's why the first thing that will come to your mind, so the description of this is that this one contains additional nutrients, okay, that you can get from blood. Blood is rich in a lot of nutrients. So when you enrich your nutrient agar with blood, that is an example of enriched media. Sometimes you also enrich it with serum. Sometimes you can enrich it with egg. Sometimes you can enrich it with other things. So enrich with this to support the growth of fastidious, fastidious organisms. Okay, fastidious organisms. So fastidious organisms are organisms that required special treatments. That's just the way, that require special nutrient, or they require other special extra nutritional support. Okay, nutritional support. And whenever we mention this, the two things that will make you, the two examples that will come right to your mind is a blood agar and chocolate agar. The difference between blood agar is that you prepare normal nutrients agar into the flask, but while it is still very, very hot, it's still going to be in liquid form. By the time you weigh the powder, they ask you to dissolve 28 grams of this powder. You dissolve it in a 100 1000 milliliter, that is 1 liter. You dissolve 28 gram in 1 liter. So depending on the one you now need, don't forget that if you are going to prepare, this is petri dish. Under normal circumstance, in this petri dish, the liquid agar that you should put there should be between 20 and 25 ml. 20 and 25 ml. That means, because the instruction here is that you will have to dissolve 28 gram, in 1000 milliliter. So if you are going for the maximum, so how many 25 will you see from here? So you are going to need 40. Okay, 40. That means you will need, if you are making use of 28 gram and you want to prepare 40 of this, that means you will need 40 petri dishes to be able to take care of that. So that is what is going to determine how much of this you are going to weigh. So when you dissolve 28 gram in 1000 milliliter, so if you are going to, so that means 14 gram is going to be for 500 milliliter. Half of 14, that is 7 gram, we go for 250. So, and that 250 will mean that you are going to have 10 of petri dish. So for 10 petri dish, you will weigh 7 gram of this. And don't forget that the whole content of this is 500 gram. 500 gram. So you can now calculate how many 28 gram can you see from 500 gram. That will tell you how many times of this 28 you take from the whole of this. Okay, so that is very, very important, and that is how to calculate your working solution. So by the time we actually start the practical, you are going to see more of this. But one thing you should have in mind is that whenever you want to prepare blood agar, you will collect blood. You can make use of already expired human blood, or you can collect fresh blood, but you can also collect blood from sheep and from animals. Okay, but the only thing is that you want to grow microorganisms that will require extra nutritional support or extra nutritional treatment. So that is that. So, and that one, you will use it for the growth of fastidious. When I say fastidious, it means microorganisms that require extra nutritional support. So while you prepare, so you are going to prepare this. This is so this is your liquid molten, because you will autoclave it for at 121 degrees centigrade for between 15 and 20 minutes. So thereafter, while it is still very hot, you add the blood. When you add the blood is going to lyse and it's going to change color. So that is for chocolate. That's why they call it chocolate. But when this is as cooled to between 40 degree and 45 degrees centigrade, or even below 40 degree, less than 40 or maximum of 40 degrees centigrade, that is when you add the blood. When you add the blood, because it is no longer hot, that is telling you that the color will not change. That blood color will not change. Now the question is, what are they looking for? What are the microorganisms? What are the basic interests that microorganisms will have so that they want to consume blood? Don't forget that inside the blood, we have an iron loving molecule which is called hemoglobin. Hemoglobin. Okay, microorganism that actually want to metabolize hemoglobin, they are the ones that are called fastidious. They are the one that requires enriched media. You need to enrich their media. So do you know that there are some people that ordinarily they can't take gari, they cannot take gari. Some of them will not even take akamu, that's ogi, unless they have other things that they want to enrich. So the same thing. By this microorganism, they are interested in the hemoglobin. And why are they interested in the hemoglobin? Because of the iron loving substance that is present in that. They have a specialized enzyme. They contain fastidious organism contains an enzyme called hemolysin. Hemolysin. So it is this hemolysin that is going to lyse, that is going to combine with hemoglobin in order to break it down. So that is what is called what? That is those that will require special treatment. Those that require special treatment. Okay. So I want to give you time briefly so that you'll be able to copy what we have under. But don't forget that a nutrient agar, the purpose is a broad spectrum, and it is used because of their simplicity. Anybody, general purpose. You are not interested in identification. You are not interested in detail. What you are interested in is just want to grow microorganism. It could be, it could support even the growth of fungi. So because of their simplicity, people, this is the starting point. This is the starting point of your journey as somebody that want to cultivate. You just want to cultivate microorganism. So your mind should go for that nutrient broth. That is, if you are working with liquid. If you are working with liquid like blood, you are working with cerebrospinal fluid or other. So what you make use of is just a nutrient agar and nothing more. So the next one, the next type of agar is what is called enriched. Enriched agar. Just from the word enriched, that means you have ordinarily, you have fortified, you have reached, and you have enriched, you have added an additional nutrient apart from the normal one. So you have, apart from somebody that's going to take gari, so you have added sugar, you have added milk, you have added moi moi, you added granules, you added a lot of other things. So for you, so an example of that is a blood agar as well as a chocolate agar. And I told you that what's the difference between blood agar and chocolate? The difference is that chocolate is being added to the molten agar while it is still very hot, so that the very hot molten agar is going to change the color. It's going to split it. It's going to change it to chocolate color. So, and that of blood, you had the blood when it is still very, the temperature has actually decreased. And I told you that nutrient agar, on nutrient agar, the direction is that you should use 28 gram in 1000 milliliter. And each of the flask, each of these petri dish should contain, okay, this is a petri dish. This is a petri dish. Inside here you have 20 in a row. 20 in a row. So that is telling you that if you are going for, and each of these should contain nothing more or nothing less than 25 ml. That is telling you that if you are truly weighing 28 gram in 1000 milliliter, that means you need 40 of these. So that means you are working on something that you need 2 rows of this one. So if you need 2 rows, that is when you are going to make 28 grams. But if you are working with just 1 row, that is half of this, which is going to be 14 grams. So for you to be able to use the whole of this 20 to be able to prepare your nutrient agar, you need just 14 grams to weigh 14 grams. And when you weigh that 14 grams, that means it is going for 20. So when this one is going for 20, so if you want to make use of 10, that means you weigh 20 grams. So that is how to be able to prepare your working solution in the laboratory. So then the second one, just like I told you, is enriched media. So, and we are going to see this as we continue. So I want to really believe that at this point, let me just go to one more. Let's take one more so that it is not going to be too much for you to be able to assimilate. And that one we are going to pick is number 3, which is a selective. From the word selective. So this type of media is very, very selective in nature. Very selective in nature. And I'm going to give you examples, just like I gave you in the case of general purpose. So let's look at the third one, which is selective. Okay. From the word select. So you must have added something that is going to select. So the third one is selective. Selective media. Okay. Now, just like I said, from the word select, so that means it has contained an additional thing. It has contained an additional material. It has contains additional, called additives. Okay, additional. So like you have garnishing of soup. That is selective. And in this one, not everybody that do like all these garnishing reagents in their, okay. So addition, such as antibiotics, that contain some antibiotics, some dyes, such as crystal violet. Crystal violet will prevent the growth of gram-positive. Okay. Crystal violet. Apart from crystal violet, some of them, they must have added sodium chloride also to be able to also do this. Okay. So this one, the additional materials into this will prevent the growth. That's why they call it selective. You are selecting microorganisms. You are interested in some other microorganisms while you are at the expense of others. It's just like in the whole class now, out of all the people that knows how to take different rice, you now want to take those ones that we will be able to take that green rice or fried rice or what have you. You still add some other things that other people might not be able to even like at all. So that is the same thing, the same principle. Select. So you select the growth of others while you deselect the growth of others. So in this case, you are selecting. Okay, example. Let me give you examples. There are different examples. So example is our McConkey. Example is McConkey agar. Okay, this is very, very common. So this McConkey agar, anything that grows in McConkey agar, you don't need anybody to tell you. Any microorganism or any bacteria that grows in McConkey agar, because they have incorporated antibiotics as well as a crystal violet. These two will not allow gram-positive to grow. That means any microorganism that grow or any bacteria that grow in McConkey, something should be telling you that must be gram-negative. That must be gram-negative. So if you are interested in gram-negative, knowing you want to know or study the behavior of gram-negative bacteria, so you should just go and get agar that is called McConkey. So it is that because it has a crystal violet as well as some antibiotics that will prevent the growth of gram-positive. So majorly what will grow there is going to be gram-negative. You can see now that it is selecting for gram-negative but deselecting gram-positive. Okay, so you need to know this. So another one, I don't have McConkey here, but another one that I have two here that you will know. Another one is a mannitol salt agar. Mannitol salt agar. This one is specifically used to grow staphylococcus. So if you are interested in, if what you are looking for is the staphylococcus, you'll be able to use mannitol salt agar to be able to prepare that. And this is one mannitol salt agar. Okay, you can see mannitol salt agar, the same principle you use in preparing other agar like a nutrient agar, the same principle. And let me just read out some of the ingredients there. The ingredient has a sodium chloride. You know, I told you sodium chloride. So this one will prevent the growth of a gram-positive, and that's the reason why when we get to preservation techniques, you see that if you don't want bacteria to grow on your iru or your meat, what you do is to add salt, sodium chloride. And that's the reason why you also had, apart from sodium chloride serving other function, it also eradicates or kill gram-positive. That's why you also had it. That's the microbiological principle. Why we normally had, if you don't want anything to grow bad, you had salt, including our soup. So the salt therein is going to certain level, is going to inhibit, prevent, or slow down the activities of gram-positive. So mannitol salt agar, it is used to grow staphylococcus species. And apart from that, we have a D-mannitol. Mannitol is a sugar, is a carbohydrate. So, and this one says you will dissolve 1 gram instead of 28 gram that you have in nutrient agar. This one says you should dissolve 111 gram of this in 1 liter of that. So another one, highly selective is SSA agar. That's Salmonella shigella agar. That is another selective agar that you can use. So this one from the word, this one can only be used to grow Salmonella species and Shigella, especially from clinical and non-clinical samples. Okay, so it's called SSA, that's Salmonella, Shigella, agar, and the short form is SSA. So this one is used to grow Salmonella. Okay, and why are we so interested? So that is the purpose. The purpose of using selective agar is more of identification. It is at this point that you want to identify. You want to know selective, your selectivity. By the time you are selecting, you are already differentiating, you are already distinguishing, you are already discriminating. So, and that is the number one reasons that is going to tell you which of these agar that you should use. If you are looking for staphylococcus, you will, if what you are looking for is just gram negative, especially lactose fermenters and non-lactose, which is gram negative, you use McConkey. If you want to know whether the gram negative that grows on McConkey is a staphylococcus, so that means you will just use you prepare mannitol salt agar, so that from here you just pick from McConkey, you now inoculate it in mannitol salt agar. If there is any growth, that will tell you that, oh, it is a species of staphylococcus. So, and if you pick from McConkey and you are using SSA, that is, you want to know whether it is a typhoid, or maybe it is a Salmonella, or it is a Shigella that is actually growing there. So that is where I want us to stop this afternoon, because of our time, so that you'll be able to go through them and you'll be able to read and understand. But in the pre-recorded video that I told you about, I only demonstrated and I show you some of these basic things that you will require before you cultivate microorganisms. So if you are looking for any microorganism, you just want to study, you just want to see the way they grow, so your mind should be towards general purpose agar, which is a nutrient agar or nutrient broth. So if you want to look at, maybe you are looking for fastidious microorganism that require extra nutritional support, you grow them in either blood agar or chocolate, which is under enriched. And if you are interested in gram negative, strictly gram negative bacteria, what you make use of is McConkey, that is selective. And there are different types of selective agar. I told you there are some that are selective for Pseudomonas, there are some that are selective for Salmonella, there are lots. So depending on what you are actually looking for, that is going to tell you, going to prompt you the type of agar you are going to make use of. So once again, don't forget that this is Omega Bioedutainment educational series, and today again, this lecture is actually devoted for new breed in microbiology. So those that will have their career in microbiology, this is where the first general microbiological course that they need to do. So don't forget to subscribe, don't forget to like, don't forget to drop your comments. Drop your comments or feedback in the comment section. And again, just like I told you, there is going to be some other quiz that will also still come up. So make sure you check the comment section. If there is anything that you don't understand, let me know. Thank you for your listening attention. Thank you.